Good day, it is my great pleasure to present our research today. The title of our manuscript is Interactions between Parabacteroides goldsteinii and diet ameliorate colitis in mice via regulating gut bile acid metabolism. IBD, primarily including ulcerative colitis and Crohn's disease, is a chronic condition characterized by recurrent episodes. It has a higher incidence in Europe and North America, with its prevalence increasing annually in China. Studies have shown that IBD is a significant risk factor for colorectal cancer, with IBD patients facing a higher cancer risk, a younger age of onset, and poorer prognosis. The exact pathogenesis of IBD remains unclear, and its influencing factors are complex. Among these, diet, the gut microbiota, and their metabolites play crucial roles in the development of IBD. Let me introduce the main findings of this study. The relative abundance of Parabacteroides goldsteinii is significantly reduced in the feces of IBD patients. Animal experiments demonstrated that oral administration of P. goldsteinii can alleviate DSS-induced colitis and AOM/DSS-induced colorectal cancer. Intervention with P. goldsteinii improves DSS-induced intestinal metabolic disturbances, particularly bile acid metabolism. Diet influences the colonization efficiency of P. goldsteinii and its anti-inflammatory effects. Next, let me present the results. First, we found that the abundance of Parabacteroides goldsteinii was significantly reduced in the feces of IBD patients from both the United States and China. Furthermore, using the Gene Repo database, we observed a significant reduction in the detection rate of P. goldsteinii in patients with intestinal inflammation. LEFSA analysis indicated that P. goldsteinii is a characteristic differentiating species between healthy individuals and those with intestinal inflammation. Next, we explored the effects of P. goldsteinii on colitis using a DSS-induced mouse colitis model. The results indicate that, under a fiber-free diet, P. goldsteinii provides significant protective effects in mice with colitis. IBD is an important risk factor for the development of colorectal cancer. To further validate the anti-inflammatory effects of P. goldsteinii, we next examined the impact of P. goldsteinii administration on the AOM/DSS-induced colorectal cancer model. The results showed that, compared to the model group, P. goldsteinii intervention effectively suppressed inflammation-associated carcinogenesis. The main way in which the gut microbiota interacts with the host is through metabolites derived from the microbiota. The untargeted metabolomics results showed that P. goldsteinii intervention partially improved the metabolic disturbances in the feces of mice, particularly in bile acid metabolism. Subsequently, the targeted metabolomics results indicated that, regardless of the diet, secondary bile acids were significantly reduced in the feces of mice in the model group. However, only in the FFD group did oral administration of P. goldsteinii significantly increase the levels of secondary bile acids in the feces. Given the close relationship between bile acids and colitis, we first examined the impact of a BSH inhibitor on the anti-inflammatory effects of P. goldsteinii. When the BSH inhibitor was used, the anti-inflammatory effect of P. goldsteinii was significantly reduced. This suggests that the anti-inflammatory effect of P. goldsteinii is dependent on BSH. Next, we found, in the absence of TGR5, the protective effects of P. goldsteinii on body weight and the colon were completely abolished. This suggests that the anti-inflammatory effect of P. goldsteinii is dependent on the TGR5. In summary, we found oral administration of P. goldsteinii can effectively alleviate DSS-induced colitis in mice and inhibit the formation of colonic tumors in the AOM/DSS model, making it a potential probiotic. P. goldsteinii exerts its beneficial effects in improving colitis by modulating gut bile acid metabolism. Diet may influence its anti-inflammatory effects by affecting the colonization of P. goldsteinii and bile acid metabolism.